Okay, I reckon it's right on 12 o'clock. I think we should kick this off and people can join us as the time goes on. So before we start this presentation, I want to let you know that QENDO acknowledges the traditional custodians of country throughout Australia and their connections to land, sea, and community. We pay our respect to past, present, and future people who have walked and cared for the land, and we acknowledge and respect their continuing culture and their contributions to make this life of this country. We also acknowledge those with a lived experience and their significant oh my goodness, and their significant others who walk this journey with us. So I'm absolutely thrilled to join with us today for this special hour with QENDO, Professor Joel Castro. Now, Joel, I will give you a little bit of a bio, but if you want to give a little bit of an extended one after this, that'd be great. So, Joel studies mechanisms of chronic pelvic pain and endometriosis and other disorders such as IBS, IBD. Now, Joel has also developed two animal models of endometriosis to investigate changes in sensory pathways. Dr. Castro has also published over 60 papers. So that is really, really cool. I'm really looking forward to this hour with QENDO to learn more about the research side and chronic pain. So without further ado, Joel, I'll hand over this session to you. And if anyone has any questions, we do have the Q&A open. And what we'll do is we'll run through the session and then at the end we will have a question and answer time. So Joel, over to you.
Thank you so much, As. How are you all? And very good afternoon. Thank you to join us here in this Hour with Kendo session. So today I'm going to talk a little bit about the chronic pelvic pain in endometriosis. That would be from the perspective of researchers. That's who I am. So I'm not a clinician, I'm a researcher, but I work in the South Australian Health and Medical Research Institute, or SAMRI, here in Adelaide. Within the visceral pain group, I have the head, I lead the endometriosis research group. We've been doing research in this area for the last 5 to 6 years, and I'm pretty excited to talk to you a little bit more about it today. So I kind of had a structure the presentation that I can talk to you about a general background in just talking a little bit about the pathogenesis of endometriosis, its impact, and the current treatments that are in there for it. About the type of pains, specifically the pain in endometriosis, and about what is limiting the developing of new and effective treatments for pain in endometriosis, and what are we doing as a research group to fill this gap and ultimately improve the quality of life of patients that have endometriosis. Right. So first, a little bit of the background. You may know, but I tried to put like a simplified way of what is the pathogenesis of endometriosis. Basically, this disorder occurs when there are cells that are similar to those lining in the uterus are found in other parts of the body. So there is the normal cycle growth and maintenance of the endometrium, which is the layer that inner layer of the uterus, and that is being expelled during the normal menstruation. But in some cases, there is this phenomenon called retrograde menstruation, in which these endometrial cells migrate outside the uterus into the peritoneal cavity. And wherever they land in the different internal organs within the peritoneal cavity, they form these lesions, these endometriotic lesions. Now, the impact of this disorder is huge. It's estimated that affect 1 in 10 women worldwide. And here in Australia, the numbers are even higher. It's like 1 in 7 women are affected by this disorder. Unfortunately, 80% of women that have endometriosis experience chronic pelvic pain. And that comes in a variety of ways. So, last menstrual cramps, pain during or after sex, intestinal pain, and also pain related with bowel habits like defecation and urination. The other big symptom that these patients have is infertility. Around half percent of the, 50% of the women with endometriosis suffer from infertility. Now, staggering there is a delay of around 7 years after the first symptom to show up in these women for them to be diagnosed that they do have endometriosis. And this is mainly given because of the traditional way that the endometriosis is being diagnosed, which is through a surgical laparoscopy and, seeing what's happening inside the peritoneal cavity. I do might say though that there is a big shift nowadays in terms of using more revolutionary and non-invasive approaches, like a combination of ultrasound, MRI, and artificial intelligence, just to analyze these images and understand if there is the presence of endometriosis at a very early stage. A lot of people are moving into this space, including local clinicians here in Adelaide, Louise Hall and her crew, using this fantastic program called Imagiendo, and also in other parts of the world. But this is great because that will allow an early diagnosis of the disorder and therefore the possibility to do an early intervention. So the quality of life of the women that have endometriosis is really affected. There is huge school avoidance, absence, loss in productivity, social avoidance in general, and the aspect of the day after day life, like the sexual function, is being affected, which bring relationship issues, and basically the mental health and the well-being of these women is really deteriorated. And of course, this brings a huge economic burden from any economy. Just Australia alone, there is like around $10 million that the government, spend in a year trying to manage this disorder. So the current treatments that are in there, there are different type of lines of treatments. The first one would be pharmaceutical treatment. The hormonal treatments, basically the pill, because this disorder is an estrogen-dependent disorder for this lesion to form the higher level of estrogen. So if you provide the pill or the hormonal treatments, you prevent for that to happen and therefore for this lesion to develop. But this is not good because it prevents women from conceiving. So it does relieve the pain, but it affects young ladies that want to have a family, and that's not ideal whatsoever. There is also non-hormonal treatments like anti-inflammatory, non-steroidal anti-inflammatory, and something stronger like opioids. But again, they are no effective and long-term use of these drugs, especially like the opioids. They cause dependence and actually other side effects that increase chronic pain, chronic pelvic pain, like constipation, for example. And unfortunately, if you stop taking these treatments or tablets, the pain will reoccur. Now, there is the option of remove this lesion through surgery. This is most invasive to treat pain and infertility, and often the full removal of these lesions is not 100% effective, and pain remains or come back in approximately 40% of the patients that they undergo this surgical procedure. Finally, there is even more drastic way of surgery, which is the removal of the whole uterus, and sometimes that comes with the removal of the ovaries as well. This causes early menopause, which actually is normally treated by hormone replacement therapy, but that by itself can exacerbate chronic pelvic pain. So it's by far ideal, and also there is a lot of cases of recurrence even after this drastic way of solve the disease with this drastic surgery. So there is no doubt that we need non-invasive and non-hormonal treatments to treat chronic pain associated with endometriosis. Talk a little bit about the type of pains, because in that way we understand more what happens in endometriosis. There is the acute or the immediate pain. Within the pain scientific area, we call nociceptive pain. And basically, this is like a protective mechanism that is signaling the body to avoid further harm. So it's like an alarm for injury. Now, there is the inflammatory or the injury evoked pain. And basically, when there is an accident, the body initiate an inflammatory response to clear the debris and beginning the healing of the area that is being affected. Because of that, there is a lot of inflammatory mediators that are in that area, and they are known to act directly into pain-sensing nerves, which we call nociceptors. So that's the reason and the origin of this pain. And because of this nerve becomes sensitive to stimuli, they develop hypersensitivity, which is the scientific name for it. And basically, stimulus that is normally not painful, like just a normal touch in these areas that are, affected, have the injury, they will result in a painful sensation for the one that had the injury. And actually, the stimulus that are painful, if you bump that area, that becomes more painful than which should be to a normal people. Now, finally, this chronic pain, that's the other type of pain. This is more intense than the acute and the inflammatory pain, and it can last on and off for years. In some cases, there is no apparent physical reason for the pain. So that happen in cases like this initial injury or accident has, completely healed, but you still feel pain in that area. And it's been proposed to being originated because of long-term changes that occur in the peripheral nervous system and actually in the central nervous system. And basically, it's like an ongoing false alarm for injury or harm. These pain affect significantly physically, emotionally, and the actual social well-being of individuals is being affected when you suffer this type of pain. So what about endometriosis? Pain in endometriosis. So endometriosis is a chronic inflammatory condition. That's definition of it. And basically, as I mentioned before, once the cyclical bleeding from the retrograde menstruation occur, that initiate an inflammatory response within the peritoneal cavity. So there is an increase of these inflammatory mediators within the peritoneal fluid, which is the liquid, the bath of all our internal organs. Um, but moreover, there is an inflammatory response within the endometriotic lesions that are growing, um, and they contribute to the increase of inflammatory factors within this peritoneal fluid, which eventually acts directly into the peripheral nerve endings that are innervating different pelvic organs, including the female reproductive tract, the colon, the bladder, and so forth, and also the nerves that are within these lesions, because as I talk a little bit more later on, these lesions are innervated by nerves and blood vessels, which allow them to grow. So basically, this is what happened with the inflammation in terms of acting directly into the nerves. And when that happens, this nerve actually release further inflammatory mediators into the peritoneal cavity in a process called neurogenic inflammation. So that cycle just maintain and that cause something called peripheral sensitization. Basically, these nerves become sensitized. The activation threshold for them is reduced, and that translates that the pain is induced from a stimulus that normally don't cause any pain, and that's called allodynia. And also that painful stimulus feel a bit more painful than they should be, and that's called hyperalgesia. So basically, these peripheral nerve endings can, at the end, become chronically hypersensitive, and that happens even after this inflammatory environment just stop to occur. So as you can imagine that this happens and this happens every time that the cycle is present. So eventually the central nervous system becomes sensitized, and that's called central sensitization. So this chronic hyperexcitability of the peripheral nerves, and that provoke long-term changes within the central nervous system, within the spinal cord and the brain. And basically, this acts similar to what happened like in memory, that pain persists long after this insult is resolved, so the pain becomes chronic. This is seen in other chronic visceral painful conditions, such as irritable bowel syndrome and bladder pain syndrome. And it may explain the reason why, even after you remove these lesions, there is still pain in this patient. And it's just because this central nervous system is sensitized, so it's engaged, and there is always this painful information being sent into the brain. So there is no doubt that pain endometriosis is a very complex problem. So, and summary is like originated by a dysregulated immune system that is switch on constantly. Uh, it involves more than the physical location of the lesions, and it's driven by changes in the peripheral and the central pain circuitry. As I mentioned, it takes like around 7 years to diagnose. So by this time, these long-term changes in this circuitry has already occurred, and that resulting in chronic pelvic pain. And it's actually really hard just to reverse back. Now, in addition of all of this, if you have endometriosis, there is most likely chances that you experience other visceral painful comorbidities, like irritable bowel syndrome, interstitial cystitis, and pelvic inflammatory disease. So it is quite complex. Sorry. Now, going to talk a little bit about what we think is limiting the developing of new pain treatments for endo. So we think that the reason of this is not knowing how pain is detected and transmitted by the sensory nerve endings that innervate pelvic organs that are affected by endometriosis, such as the female reproductive tract, but also the colon and the bladder and so forth. It is also not knowing how this painful signal is then integrated and processed within the central nervous system. Now, what are we doing to help endometriosis patients? Our group basically focus in determining how normal sensation and pain is detected by these sensory nerve endings that are innervating the female reproductive tract and within the central nervous system. And actually also to determine how the molecular composition, the structure and the function of these nerves is altered by endometriosis, causing chronic pain. And finally, we try to validate how we can target those changes that we have found in this peripheral nervous system to reduce chronic pain in endometriosis without using invasive procedures or harmful side effects. To do this, we have developed a series of techniques and approaches throughout the years. We have developed, as Ash mentioned, two more mouse models of endometriosis. It is necessary in this case or in this area of research the use of animal models. Complex pain is a complex problem, and you cannot just solve it or model or understand how that happened just by using computational models or in vitro cells and so forth. You need the whole integration of what happened in the body, in this case in the animal, to understand how that pain changes occur. So we have developed, as I mentioned, a couple of animals of endometriosis. They display similar features to what the humans have, like these lesions infiltrated by blood vessels, by nerves. They have rich inflammatory environment. These animals that develop endometriosis also display a widespread pain, and that's manifested by the pain arising from the vagina, the colon, hypersensitivity of the skin, and sometimes changes in the general behavior of the mind, of the minds, which reflect pain and discomfort. We also have through our valuable collaborators, some of them here in Adelaide, like Luis, and the group at the Flinders Medical Center, and some of them in Melbourne and April as well. We have access to biopsies from women with or without endometriosis. Basically, we can study what happened in the normal endometrium of these women. And in the lesions, we also have access to the peritoneal fluid, which is quite important because, as I mentioned before, have this rich inflammatory environment on it. Sorry. More recently, we have established a collaboration with some researchers in the University of Wollongong, and through them we have access to sensory neurons that are derived from pluripotent stem cells from humans. So this is an amazing technology that has emerged in the last 5, 10 years. And basically, you can get a biopsy of the skin of a person, and through a process that involve special factors in the media, you can differentiate the cells that are in this skin biopsy into different cell types, including sensory neurons. So we can actually use all this type of human, material, valuable human material, and corroborate all the findings that we found in the mouse model. Um, just understanding if the mechanisms that we're thinking that driving pain are also present and applicable into humans, which is extremely valuable for this research. We also have a way to see the research by using a multidisciplinary approach that go all the way from the molecular components of these nerves to the whole animal. Basically, throughout the years, we have developed different techniques that allow us to measure the electric activity of the nerves that go into peripheral organs like visceral organs, like the female reproductive tract, for example. In this case, this is the female reproductive tract of a mice. And we can isolate the nerves that go through there and see how they function. Basically, we can see what is different in endometriosis, and there is a lot of difference in terms of how they function. And we can see what can we do to modulate that activity and therefore modulate the pain that is being sent into the central nervous system. We also have a battery of genetic transform animal models. For example, this one here express fluorescent molecule in the sensory nerves. Now, these are the female reproductive tract, the uterine horns, the vagina, like this, that is being open and pinning flat. And if you can zoom this image in here, you can appreciate the complexity network of nerves that are in just in that reduced area. Now, this nerve change, the structure of this nerve change in inflammatory conditions, and these changes can explain why this abnormal sensation happen, which is translated into pain, basically. We can also study the molecular composition of these nerve endings. And basically, to do that, we collect the cell bodies of these nerve endings that are in the dorsal root ganglia, and using state-of-the-art techniques like a transcriptomic analysis and so forth, we can find the genetic makeup that is formed, that this unit is formed by. We can study how that changed in the disease of endometriosis, and that basically can give us some ideas of what we can target to stop pain. You can see the activities of these nerves as well in vivo with special techniques that we do, and study what happened within the spinal cord in terms of the pain circuitry, and basically with the whole animal in terms of how they behave to different stimuli. Then we evoke a mechanical or thermal stimuli and see how they respond to it, and also how they behave normally, which is an indication of like how good they feel, or how bad they feel if they have pain or discomfort and so forth. Now, what we have learned using all these techniques and materials, we have learned that these sensory nerves within the female reproductive tract, they are tuned to detect a variety of different stimulus, mechanical and thermal stimulus of different intensities and grades. We also learned that the painful signal from the female reproductive tract is mediated by a special type of ion channel, which is expressed within these nerves. This is ion channels is called sodium voltage sensitive channels or NAV channels. And we went even further and find that if you inhibit these channels using purified spikenvenom, we can reset the abnormal sensory circuit and stop chronic pain in these mice that have endometriosis. This is a collaboration that we have done actually with some researchers at University of Queensland with Glenn King and Irina Vetter. And these are fantastic guys that they isolate all these venoms from like spiders, jellyfish, and snakes. And basically, the idea is that they have some molecules from them that directly control the activity of the nerves. They use that in their normal defense or to hands, but we can just harness that power that nature is giving us to find these compounds that hopefully can stop or modulate how the nerve just transmit the information to the nerve to the brain, and therefore we can modulate pain. This is very exciting, and maybe that can become a possible treatment for endo in the future. I know Glenn has like a lot of work with Os Health and to try to translate this research into clinical outcome. Now, we know also, we have learned also that endo alter the gene expression of the sensory neurons that innervate this organ. Basically, we found that there is huge number of genes that are upregulated by endometriosis, and they can in the future become potential targets for pain. We are currently doing a lot of work to see which they are and try to validate it. If we can just modify it, that will produce relief, at least in our model. Excuse me. So basically, what we're focused is in targeting these ion channels and different receptors that are expressed in these peripheral nerve endings, because this is the first step of the pathway of the pain. So we try to see if we can just modulate the activity of these channels, we can stop chronic pain. And that have worked, at least preclinically in these animal models. That have worked for other visceral painful disorders like irritable bowel syndrome. We have, and other groups have published some information articles that describe that this is the case. And as I mentioned as well, more recently, that the case for endometriosis. But in reality, only two drugs of the kind, or two drugs that are alter the activity of these channels in the peripheral nerves, have made it to the clinic so far. So one is ciconotide, which is derived from a marine corn snail that target a particular type of ion channel. And it's being approved like 20 years ago to treat a variety of pains. But this drug is need to be given intrathecally, so injected into the spinal cord, and that has huge side effects potentially within the central nervous system. So it's not an ideal. Now, recently, and this has caused a lot of attention in the scientific community, there is a new drug in the market now that is being approved at the beginning of the year called Suzetrigine. And this is a synthetic origin drug, and it is being used to target NaV1.8, which is another type of NaV sodium voltage sensitive channel that are expressed in the peripheral nerves. Is being recommended to treat moderate to severe acute pain. It's not there for endometriosis yet, but we are all hoping that, that can be extended to that application. But the reality is that so far there is no an improvement drug that use this strategy to treat the endometriosis. We are working on it. Hopefully we can, change this and try to contribute to for this to happen. Now, in the last part of the talk, I'm going to talk about something a little bit different, and it's like, what other things are we doing as a group to relieve the burden of endometriosis? So, what about preventing the lesion development as such, as a treatment? Now, these lesions are not to develop, as I mentioned, blood vessels and nerves, and that allowed them for them to grow. This process is called neuroangiogenesis. We also know that the peritoneal fluid within the peritoneal cavity and within the lesions is rich in these pro-inflammatory mediators or factors, and they promote this process of neuroangiogenesis. But they also promote other processes within the lesions that control the establishment of them and allow them to grow. And on top of that, we know that they act directly into nerves, just producing pain. So what about if we can target this immune system and the process of neuroangiogenesis to stop the developing of the lesions and therefore to relieve chronic pain in endometriosis? Now, we are currently evaluating such a possibility. We are evaluating a new immunotherapeutic approach that hopefully can inhibit the growth of the lesions and the developing of chronic pain in humans. So for, I will just mention what we have done with our animal model of endometriosis and the results that we have got so far with this drug. This is a monoclonal antibody called NP137. And basically, it targets a protein that is called netrin-1, which is a secreted protein that regulates a variety of very crucial processes within the body, including the migration of these immune cells and the growth of the axons and the blood vessels. So as you can see, they could be really potentially important to treat endometriosis lesion progression. Like 2 or 3 years ago, there's a group that showed that this protein, netrin-1, is being found elevated in the blood and within the lesions of women with endometriosis. Now, this antibody, NP137, that targets netrin-1, has been shown that is effective to treat the growth of endometrial cancer or these endometrial tumors. And that's been shown to happen in animals, but also in humans. So it's in actually in clinical trials for 3 years already to treat endometrial cancer. It has been shown very good safety profile and actually being effective to treat this type of advanced cancer. Now, I should mention that the lesion growth, the growth of these endometriotic lesions, is similar to the process of tumor formation and growth. They're very different processes, but the actual things that make them grow is similar. And that is because of they both are regulated by the immune system. So in both of them, there is a dysregulated immune system and there's the development of neuroangiogenesis. So they make it like really similar. Therefore, we believe that we can use this antibody that is known that work in cancer. We can actually find a way to reduce the growth of the lesions in endometriosis and also the chronic pain. So this happened as like 3 years ago with a collaboration with the French group that actually was the one that discovered the netrin-1, this protein. And have done an amazing work in describing how it works in the body and the processes that it regulates. They had created this monoclonal antibody, and we got in contact and they say, you know what, I think this can work for endometriosis. Are you keen in trying? And say, yes, of course. So I'm going to talk a little bit about the rest of the talk of what we did, which is something that is pretty exciting, and we are really in the middle of it now. So basically, we found that these antibodies reduce the lesion growth in our mouse model. This is like a scheme just showing what happened in terms of the study. So we have sham or control or healthy animals and animals that have endometriosis induced by the way that we have established and characterized before. And like about 4 weeks after, when we know that these lesions are roughly half developed, we treat these animals with the monoclonal antibody or with a control monoclonal antibody. 4 weeks later, we then do a phenotypic assay. So we see the pain that this animal experienced through different techniques, which I'll show you later on. And then at the end of that, we collect the lesions and we measure the size just to see if the treatment reduced both the pain and the development of these lesions. So it does. I'm showing you these are representative female reproductive tract from CHAM mice that are being treated with the control antibody. They, look normal. And this is how the female reproductive tract of mice that have endometriosis look like. These are huge lesions that they develop around in the places that we did the surgery for that to happen. There's also a huge develop of fibrosis within that area, and all of that are common things that you can see in women with endometriosis. Now, if we treat these animals with the MP137, there's a marked reduction of these lesions, and you can see if you excise these lesions out of the organ, the different sizes is striking. We can quantify this reduction by measuring the weight of them and the size, the diameter, and you can see the monoclonal antibody have a dramatic effect in reducing their growth. So this is really exciting about these results. Now we also found that it does reduce chronic pain and also all the comorbidities that are developed in this mice model. When we treat these mice 8 weeks and after 4 weeks of the treatment, we find that the enhanced sensitivity that mice with endometriosis show when you distend the vagina or the colon um, is reduced by the treatment. So basically, this is a measure of pain that the animals perceive when you distend hollow organs like the vagina or the colon. These are animals that are healthy or sham animals, so they respond upon increasing distension pressures of this organ. That is enhanced or exacerbated in endometriosis. That show that there is hyperalgesia, so they have pain when you distend that area. Now the treatment group, which is in green, you see that it's closer to the healthy, so there's a reduction in the pain that they experience if you distend that particular organ. There is also a reduction of the sensitivity of the skin to different stimuli, to the thermal stimuli or to mechanical stimuli, which we do that by probing with a very fine filament in the abdomen. So these mice that have endometriosis, they have a reduced reaction to this stimulant, so they are uncomfortable. They are sore in the areas that the stimuli occur in endometriosis, and this treatment just reverted. It also reverts the bladder dysfunction that mice with endometriosis develop. And we see this because of if mice have, sorry, if there is dysfunction of the bladder, the normal organized voiding pattern of the mice happens in each of the cages. But if there is a dysfunction of the bladder, if there is pain in the bladder, this gets disrupted, and basically the animal just like we scatter it all around the cage. You can quantify the number of spots, as you can see in here. Uh, with the development of endometriosis, there is an increase and the organized voiding pattern, which is indicative of pain. And the antibody just correct that, just normalizing to what it looks to a healthy animal. Finally, we also measured the spontaneous behavior. So how the animal just go about it with their normal life. For this, we do this nesting building assay. We basically give to the animal put in the cage a nest of material, which through the night they just shredded and they form this beautiful fluffy nest that they use to be comfy and get protected. Now, if the animal doesn't feel good, if they have pain or discomfort, they don't do this. They just leave the nest all halfway done. This nice fluffy nest can be formed. They don't spend the time doing it. And this is exactly what happened with our mice with endometriosis. As you can see here, there's a reduction in the nest and building capacity. And if you treat these mice with a control antibody, that is reverted. So it looks that the animal feels much better. Basically, this is what we have done so far in terms of testing that this antibody work, and the results look that it does. It look that this is a feasible therapy that is non-invasive to treat endometriosis developing and associated chronic pain. We are currently investigating the mechanism of action, so how to explain the beneficial effects of this antibody. In endometriosis, and excitingly, our collaborators in France, who I mentioned, developed this antibody, gearing up for a clinical trial for endo that hopefully happen next year. So I think that's it for me, and hopefully I can was able to show what we are doing in terms of our lab to try to stop pain and endometriosis development. I really thank you so much for listening, and also thank you to all the people that helped me with the work, the people from my work, from my group, especially Jessica Madden, which is with me side by side, just doing the model and all these spectacular tests. And our different collaborators that provide the venoms, the antibody, and the fantastic clinicians that we work with, and of course the funding body. And thank you again.
And Joel, thank you so very much. That is so insightful. You know, you always hear of people testing on animals, and it's always like, but you don't ever really get to see much about it. So, it really spoke to me about the mouse that had the endo, in the, like, they're not wanting to set up their bed. They're well,
Mm.
obviously tired and fatigued. So that's so very interesting. We do have a few questions and comments that have come through. Now, we might be able to answer them here. We might not. So, Joel, if you don't feel comfortable in answering them, let me know and we can take them offline and we can sort of work out some comms to share with you guys post session. So,
Perfect.
the first one is, what's the degree of success of the patients that undergo surgery and then follow up with hormonal treatment?
Yeah, so there is a degree of success, and that varies between individuals. I'm not a clinician. I'm more a researcher, so I really know I don't have all the statistics and the number just to say a precise number. I know by general experience and colleagues and people that I know that some of them there is a huge help and some of them don't, or they are still in the process just to look for an alternative that fit to them. It's very complex the process, and as I explained, what happened is about this central nervous system is being compromised already, so you can have the surgery and sometimes that help completely and it sometimes doesn't. And the thing is just look for alternative, maybe look for another opinion, someone that have a different perspective. I mean, this is just my advice as a person, as a scientist, not as a clinician. But yes, it's not 100% effective, uh, and it varies within person to person.
Yeah, person to person. Endo is
Mm.
so person specific that what might work for one
Mm.
person might not work for the other. The
Mm.
next one, could you please explain further how Journavax works and how it could be helpful for endo pain?
Yeah, so the nerves that innervate the peripheral nervous system function through different proteins or ion channels. So basically, they are the one in charge to propagate the actual potential, and that's how nerves communicate, or every signal from pain to normal sensation is being sent into the brain. That's electrical signal that happens through the nerve is mediated by these ion channels. This drug inhibit that ion channel. An ion channel that is key for this process is called a voltage-gated sensitive channel that basically allow the depolarization of these neurons and the formation of this actual potential. So it actually target the first step of the pathway, which is the nerve, or the pain pathway that is the nerve. So by doing so, the theory behind is like all this constant painful signal that is being sent to the brain stop, and then there is a time for the circuit to remodel itself. Um, so yeah, I guess we will need to see what happens. It's relatively new in the market, and I know there's a lot of attention to it. It has very good safety profile, and yeah, I'm really hopeful that can be extended to endo. So maybe it is already, and I don't know. Again, I'm not a clinician, so I know, in the core of it. But yeah, it's a very promising because of it target the sensory nerves in the peripheral nervous system. It will avoid complications within the central nervous system, like the opioid do, because the opioids act at the level of the central nervous system, and that is what causes addiction and dependence and so forth. So it could be really good.
Thank you. This might be one that you're able to help with. Is it scientifically established that endometriosis recurs after excision surgery if people are using hormonal management? Coming from not from a clinician side, from like a purely
Mm.
research scientific side, is that too tricky to answer?
So if you remove the lesions and then give the hormones, yeah. So I guess the actual definition of endometriosis is the formation of these lesions. If the lesions are not there, and if there's no hormones that control the growth, if you take them all and then you give the hormones, you cannot form more. So no more lesions should be formed. And that happens when, the women normally reach the menopause. The estrogen, high levels of estrogen are not there, or when you are pregnant, and then these lesions cannot be formed anymore. So, but that doesn't mean that pain is not there. Pain can still happen because of, as I mentioned, this central nervous system is being primed and wind out and like all remodeling for that to happen. So I guess eventually that will heal down, or that will just like, revert to normal. But yeah, so I guess like the answer is yes. If you remove the lesions and you give hormones, normal lesions will be formed. If you remove them all, which is difficult, now that doesn't mean that you will not have pain anymore, because there's like damage that happened already, and that could be at the level of central nervous system, but also could be about fibrosis. There's a lot of fibrosis that is happening in endo that can actually distort the nerves and just pull them back, the organs, and pull them back, and that causes a lot of like physical damage that is beyond the lesion. So it is a complex thing for sure.
Um, Now we do have a few more questions. There is one of our volunteers on the line. Han, I see your questions. I'm going to take those offline to Joel because they're a little bit more in depth. So, Joel, I'll share those with you and then I can share around. But to finish off the session, I have two incredible questions for you as a researcher. So they've come from the group and they want to know for first, what developments in endo research are you most excited about? And the prong to that is, what frustration do you experience? And is there anything that we can do for public attention to help drive that change?
Good ones.
They are good ones.
They
Thank
are
you,
good
Elizabeth,
ones.
for those. They're great.
Yeah, they are good ones. So I guess like research is like a needle in a haystack. So we kind of like funnel in a particular area that we are interested. Of course, with the normal awareness of what's going on around in the general field. In the area that I work, which is the chronic pain, which is what I'm interested on, I'm really excited to this type of approach of like inhibited this initial step of the pathway. I think it's something that I haven't seen before, or haven't been explored before. I came from an area of the visceral pain in the gut, and I kind of like applying the set of concepts and which include these ones that are quite developed in this area to endo. And I think it's going good, and it's making people aware that this could be a way, and it could be an efficacious way. So I'm excited about that and contributing myself to develop that, hopefully as a treatment on the basis that we provide a lot of information of how these mechanisms happen. And then if you know what happened, then you can stop it. But the point is like there's very little know for endometriosis especially. about how this pen circuitry is being changed. And um, yeah, but once you know, and that's what we're trying to do, then there could be alternative how to stop them. Frustrations, I try not to have to be optimistic. I guess frustration is I like to spend more time doing the research, and sometimes I have to spend a lot of time doing the admin and the difficult job of getting the funds to do this research. I think I'm not alone. Any scientists go through the same issues. Um, it's part of the system. We have to do it, but it is frustrating, and it could be not only life changing in terms of the scientists as such, but the research field changing. There's a lot of people that I know that cannot continue or that had to modify the research that they do, although they think that is interested because the funds are not there. Um, that I think is the only thing that frustrates me. Apart from that, it's an amazing environment and been doing this forever, and it's so much knowledge, it's so rich, and I think we can contribute as much as we can, in the chain of life, finding a cure, and um, that's what motivates me as well. Frustrations apart.
That is so very fair. I see in your photo there you've got Louise and Jodie as well. So that
Yeah,
is really cool. We've got a long
yeah.
friendship with Jodie and Louise as well. So that's lovely. So, everyone from here, if you're wanting to watch back the video, we will have it available in a few weeks on our Manage Endo platform, and we will also circulate the Q&A questions. The ones that Joel can answer, I will send them out to you via email so you can have a look through. But please remember that if you are needing any support, any guidance, anything at all, QENDO is here. We have our hotline, we have our mentor program, peer support groups, and QENDO Care. So, should you need any help, guidance, support, anything, we are here. And on behalf of the endometriosis community, Joel, I want to thank you so much for sharing about your research, sharing about your passion, and taking the time to present this session for us here. It's really interesting how far it's come and where it's yet to go. So thank you so very much. And I hope everyone has a wonderful afternoon, and we'll see you next time at An Hour with Quento.
Thank you so much, Ash, and everyone for your time. Thank you.
Thank you, everybody.
Should I stay in line?
Okay, I reckon it's right on 12 o'clock. I think we should kick this off and people can join us as the time goes on. So before we start this presentation, I want to let you know that Quendo acknowledges the traditional custodians of country throughout Australia and their connections to land, sea, and community. We pay our respect to past, present, and future people who have walked and cared for the land, and we acknowledge and respect their continuing culture and their contributions to make this life of this country. We also acknowledge those with a lived experience and their significant oh my goodness, and their significant others who walk this journey with us. So I'm absolutely thrilled to join with us today for this special hour with Quendo, Professor Joel Castro. Now, Joel, I will give you a little bit of a bio, but if you want to give a little bit of an extended one after this, that'd be great. So, Joel studies mechanisms of chronic pelvic pain and endometriosis and other disorders such as IBS, IBD. Now, Joel has also developed 2 animal models of endometriosis to investigate changes in sensory pathways. Dr. Castro has also published over 60 papers. So that is really, really cool. I'm really looking forward to this hour with Quendo to learn more about the research side and chronic pain. So, without further ado, Joel, I'll hand over this session to you. And if anyone has any questions, we do have the Q&A open. And what we'll do is we'll run through the session, and then at the end, we will have a question and answer time. So, Joel, over to you.
Thank you so much, Ash. How are you all? And very good afternoon. Thank you to